Welcome to Living Your Best During the Pandemic with Advanced, Metastatic, Chronic, and Non-Curable Cancer, supporting the quality of life throughout the cancer journey. Today's topic is clinical trials for advanced cancer, hope for you, hope for others, with Dr. John Mackey. My name is Nikki Fair, and I'm so pleased that you could join us for tonight's speaker. We acknowledge that we live, work, and play on traditional territories in Alberta of the First Nations Treaties 6, 7, and 8, Métis and Inuit, whose footsteps have marked these lands for centuries, and the gifts provided by elders and knowledge keepers. Living Your Best is a collaboration with Cancer Care Alberta, Wellspring Edmonton, and Wellspring Calgary. And I'd like to start tonight actually by introducing Roy Zinke, who is one of our Wellspring Calgary's incredible volunteers, somebody who I've come to highly regard over the last number of years. Roy has been living with pancreatic cancer as a part of his life for over 5 years now. Roy worked in senior management in Walmart for over 8 years. He has 2 incredible daughters and a wife who's also incredible, and loves to do woodworking and home building in his free time, of which I'm not sure he has much of. So welcome, Roy. We're so glad to have you, and I am going to pass it over to you now.
Well, thank you very much, Nikki. I'm very pleasure to be here tonight with everyone. And I'm going to start off with talking a little bit about my story and how clinical trials really saved my life. So I'm going to go over a little bit of what happened for my journey to my cancer, pancreatic cancer. In 2016, I was at work and feeling very sick. And prior to that, had a lot of side effects, my stomach, back pain, for probably months that I kind of ignored. But that day, it was a Saturday in 2016, I checked myself into South Health Campus, and they kept me there for 11 days and did all kinds of tests, et cetera, et cetera. Found out I had stage 4 Hodgkin's lymphoma, which, you know, I thought, okay, it's a cancer, but it's very curable, so no big deal. I got treatment for that. I had 3 treatments with ABVD, and all the cancer for Hodgkin's went away with the exception of a couple of lymph nodes that did not respond. So in March of 2017, I went in for surgery, and they cut me open and found 2 lymph nodes that unfortunately tested out to be pancreatic cancer, stage 4 terminal. And that was a pretty big blow at that time. And, you know, we sat down with our family and discussed how are we going to tackle this. And one of the ways we did it is we sent off my tumor samples to Germany, England, United States, Know Your Tumor, and a couple other places. And they all came back to the same conclusion, that I probably had maybe a year to live at best, and I better get my affairs in order. We decided as a family that we're not going to just accept that. And we start looking for clinical trials. We, after all the samples that we sent out, we found one through Johns Hopkins, and we were approved to do a testing there for immunotherapy through Dr. Lee. And she had done a clinical trial with 10 patients, and 9 patients did not respond to the immunotherapy, but 1 did. And they couldn't figure out why, so they started getting into it. And she found out that the 1 person had mismatch repair deficiencies, MSH high, etc. And with that, we found out through Know Your Tumor that I actually had those same markers. And it was just amazing because in 2017, Johns Hopkins just got that drug, immunotherapy, Keytruda, approved in the States 2 months before my diagnosis. So if it wasn't for the clinical trials at Johns Hopkins, I would not be alive today. And it's so important that people know that, you know, those options at clinical trials can really make a difference in your outcome of your treatment. and the treatment method that they're going to give you, whether it's chemo or immunotherapy or some of the new treatments that are out there now. I continue to work with many patients, mainly pancreatic cancers, but other pancreatic patients, including my brother-in-law, who has now stage 4 terminal lung cancer. And we are researching for clinical trials right now in Florida and in, I think it is England, to find out what we can get done there, because we've run out of options here. So again, clinical trials are what we're pushing for a lot of people that have stage 4 terminal cancer. And that's basically what my story is. I'm very fortunate to be here today, 4 or 5 years later, with pancreatic cancer. And I can say it's all due to, number one, my amazing daughters and wife, who were my advocates when I was very sick, and to clinical trials that have occurred down at Johns Hopkins. Again, those clinical trials are amazing and do help people's lives, save lives. So at that, I will turn it on to John Mackey.
Hey, well, thanks so much, Roy, for sharing your story. It's wonderful to see you doing so well. So my name is John Mackey. I'm a medical oncologist. I work at the University of Alberta and at the Cross Cancer Institute in Edmonton. And I've been directing the clinical trials program there for the past decade. So the reality is that I've spent really my entire career doing clinical trials. When I was in internal medicine, so when I was training to have the foundational work after medical school before you became an oncologist, I actually got interested in an idea that worked in kidney patients, people who are on dialysis. And there was a treatment that worked for controlling fluid collections called ascites that can happen in people that are on dialysis. And so because I was interested in cancer at the time, I actually took that idea and I walked across from the university hospital to the Cross Cancer Institute and ran my first clinical trial where we actually tried to treat cancer ascites with the same treatment that worked in patients with failing kidneys. And in fact, we did have some success there. And it's an interesting story because, you know, it's sort of one of these things that good ideas can come from anywhere. And you learn a lot from things that are working in other settings and bringing them and testing them in a new place. And what I've learned, you know, in the 28 years since I ran that first clinical trial was that I'm often wrong about things. And it really means that ideas that I think are for sure going to work may not. And then ideas that I think, you know, really are unlikely to help people sometimes have a tremendous benefit. And I'm supposed to be the expert in cancer who would know for sure what's going to work ahead of time. So the reality is, you know, the medical practice that we have in cancer is just not good enough. So your oncologist may be a nice guy or a nice gal, and he or she may have gone to 27 years of school like I did, and, you know, have a bunch of letters after their name. But the truth is, the reason that we have any tools to treat people is because trials have shown that these treatments, on average, help people more than they hurt people. And the reality is that the only way we're going to get better at what we do and ultimately have, you know, more effective treatments for people with advanced cancer is if we try new things. Because the state of the art in 2021 is admittedly much better than it was 10 or 20 or 30 years ago, but it's simply not good enough. And we all have, you know, friends and family members who are, you know, sick and unwell and maybe have died of advanced cancer. And I think that's a really, really difficult thing to change, but there is hope because the clinical trial process is one that is kind of self-correcting. So I wanted to share with you a little bit of the philosophy behind clinical trials and then talk about a few stories I've witnessed in some of my patients on clinical trials, and sort of talk about where I think we're going and where the most exciting areas are. So if you'll bear with me for that chat, then we can answer any specific questions you might have. So let's start first thing with the philosophy. So the philosophy of clinical trials starts with an admission of humility. And okay, I'm not a humble guy, and most physicians I know are not particularly humble people. But the intellectually honest ones will say that they don't know when they don't know. And it actually comes from the people that say they're not doing a good enough job and they don't know what the right thing to do. If you've got a group of those people that will collaborate together, then you can actually start to ask really important questions. And those questions are not the kind of thing, well, Uncle Sam was told he had 6 weeks to live from his cancer, and he went to Mexico and they cured him, and he came back and it's 10 years later and he's better. So that's not science. That's an anecdote, right? And you don't know whether Uncle Sam actually had cancer or whether it was a misdiagnosis. You don't know whether the rhubarb shakes he ate when he was in Mexico made the difference or not. There's no way to tell because it's not a formal research study. So that the basis of making an advance in cancer is actually doing a thorough study of a new idea. And that can be as simple as saying, okay, well, we have stage 4 pancreas cancer patients and we have this new drug and it works in lung cancer. So we're going to try it in 10 patients with pancreatic cancer. This is what Roy was describing earlier. And then you find, okay, well, 1 seemed to do really well and 9, it didn't seem to help at all. And then someone asking the next question from that trial, which is, well, why did Mr. X do so well when Mr. A, B, C, D, E, and F, and G didn't? And then taking that knowledge and taking it back to the laboratory, maybe understanding the biology of cancer, and in this case, it was mismatch repair, and using that as a stepping stone to the next idea. And so this is a self-correcting process that happens in clinical trials where you start from an admission of ignorance, you set out what you're going to do, and you're very strict about it. And you have an ethics committee to say, okay, Dr. Mackey, we think it's ethical for you to treat patients that you've determined are deficient in rhubarb in their bloodstream, and you're going to give them rhubarb infusions and see if their cancer symptoms improve. And they review the rhubarb concoction that I have. Is it safe to give intravenously? Is it free of germs? Is it what I tell it, what I say it is? And then you get Health Canada approval, ethics approval, and then you treat some patients. And then, you look at the results and try to understand them. So that's sort of starting from ignorance, putting a plan forward, actually executing the plan, putting people on the trial. And these are human volunteers. So when you put a person on a trial, you have to explain to them that A, you don't know what you're doing. Otherwise, they shouldn't be on the trial. If you knew that it was working, if you actually know scientifically this works, they shouldn't be on a trial. That should be standard of care. So you have to come from a position where you don't know all the information, admit that to the patient, have the patient understand that they're volunteering for potentially unknown benefits and potentially unknown harm that you could be doing them. And then agree to walk down that path of the clinical trial together and do the very best you can for that person. And it's a real contract. It's a real bond between the clinician and the patient, because you're both risking something. For a physician, it's hard to risk, the fact that you don't know what you're doing and admit it. It doesn't come easily with our training. And for the patient, of course, they're putting their life in your hands. And the amazing thing is, in Alberta, we have more patients on clinical trials than almost on a per capita basis, almost anywhere else in the world. We have a group of people who are willing to volunteer. And when they're signing that consent form saying, yes, I'm willing to walk this journey together with you, they're often saying things like, Dr. Mackey, I know you've told me this may not help me, but I know that I've got to do it because it may help someone else. And this is the really unselfish part of volunteering for a clinical trial, because many trials don't work, but the knowledge that we gain is going to help someone else down the road. So with that as the philosophical background for clinical trials, I want to share with you a couple of stories that come from, being in practice now for 28 years with cancer patients. And one lady whose name is Cecile, and she gave me permission to mention her name today, was involved in a cancer trial that I was involved in back in the late 1990s. And it was for a particular type of breast cancer. She had that particular kind of breast cancer. There were 400 patients on the trial. And she's the only one that's still alive today. And she's still getting the study drug. Now, the study drug that she got was a sort of a pill version of a drug called Herceptin. So it's a medication that affects her particular molecular kind of breast cancer. But everyone else on that trial, 400 patients, had the same kind of breast cancer. But for her, she responded beautifully. All the cancer went away. She's been cancer-free now. I've been looking after her, since 1998. So it's been, 23 years where she's on this medication and her cancer's gone. She's had a bunch of grandkids born and grown up and left the home. And, she's doing wonderfully well. And we've taken her tumor samples and tried to figure out, why did she respond so well when others, had some benefit, but not this kind of grand slam home run. And we haven't figured it out yet, but I've sent the tissue off to 3 different labs, hoping that someone will figure it out someday. Another story I'd like to tell you about is a patient of mine who had molecular profiling done of their tumor. We had gotten to the end of the road in terms of treatments that we could offer her for her breast cancer. And we didn't have any approved drugs. And we normally would say, well, I'll have to hand you off to the palliative care team because, really there's nothing here that I think I can help you with. But she had resources, and she asked me, so if you had money to apply to this problem, what would you do? And I told her, well, I would get my tumor molecularly bioprofiled, and maybe there's a weird and wonderful molecular problem with your breast cancer that has a drug that's not a breast cancer drug, some other drug that may have been used in some other setting. And we would use that medication for you. But, that medication wouldn't be paid for in Alberta. You'd have to, procure it yourself at considerable expense. Well, she actually did have a lot of dollars. So we sent off her tumor. We found what's called an actionable mutation that was probably part of the driver of her biology of the cancer. And she's on a medication that we wouldn't normally ever consider for her cancer. And she remains, doing very well. Her cancer is still there, but it's not growing or spreading. And she's been on this new medication for 2 years. So, when you actually don't know everything about a person and everything about their biology, you don't really know how well a given treatment is going to work. And it's only in the last handful of years that we've had some tools to start looking into, the drivers of a given cancer's biology, and where we can start to get an idea of what might actually help a person on a rational basis, rather than our usual thing, which is to say, well, we've treated 100 people like you and we know 30 benefit, and so we will offer you that treatment because that's what we usually do. With these new diagnostic tests paired with specific molecular therapies, we've got this new paradigm called precision medicine, where you could be much more rational and selective about your therapy. And ideally, you could find better treatments for people. So as a physician, an individual physician looking at my patient across the room, I would love to have that option for every one of my patients. But I'd like you to take a step back and think, if you're the premier of Alberta and you've got to make a decision for all of Albertans, can you really afford to pay let's say $7,000 for the diagnostic test? Can you afford to pay for the $100,000 of a drug that you wouldn't normally give to this patient because of the result of this diagnostic test? So would you want every cancer patient, and keep in mind, every year in Calgary, there's about 5,500 new cancer patients that walk in the door. So if you do the math, these are astoundingly high cost activities. And so as a Canadian, where we say we have a certain number of resources and we've got to do the best we can for the population, some of these very promising but not fully proven ideas have not been adopted as the standard of care. And the reason that they haven't been adopted is because clinical trials haven't shown that you're more likely to be alive if you do the new precision medicine approach. So let me explain what that means. So there have been a couple of trials that have been done where they've taken several hundred people and they've done all the fancy testing, given them all the fancy drugs, and compared them to a similar group of people where they just did the usual care. And then at the end of 5 years, how many are alive? And they haven't found a difference. But the trials were done with older technology and older drugs. They were done 5 years ago. That's how we have 5-year follow-up. What we're hoping is that with time and with the better tools and the better drugs, that one of these trials is going to show that clearly using these new technologies, it makes a difference to the population, not just to one patient, but to the whole group. It's a good idea for the whole group to do it. And so in Canada, we're waiting for those clinical trials to be conducted and finished and prove to the healthcare system that we are not just spending money on the occasional patient who benefits, but that we're spending money wisely and that we can prove a benefit to the entire group. So that's a sort of a philosophical underpinning behind some of these stories. And now I'd like to talk about, again, just briefly, talk about 3 areas where I think we're going to see a really interesting advances in the next handful of years. And the first one I'm going to talk about is something called the liquid biopsy. So people know what a biopsy is. It's when you often take a scalpel and cut a piece of the tumor out. So when Roy had the lymph nodes taken out, they opened them up and actually took them out. That was a biopsy procedure. You can't take a needle and stick it in and take a bit of tumor and sort of suck it out of the needle and then send it to the lab. That's a needle core biopsy. And those are the most common kind of biopsies that are done now. But what is really very promising is something called a liquid biopsy, where you just take a blood sample and you take it to the lab, and the lab has looked at your cancer before and it knows the kind of mutations your cancer has. And it turns out that all cells have a turnover in the body. They pop and they release into the bloodstream little bits of their innards. And that includes something called DNA. So DNA is where our genetic material is. And so you and I, if we did a liquid biopsy and you don't have cancer, you would see normal DNA sloshing around in your veins, and you could detect that you're not finding any cancer-related mutations in your DNA because you don't have cancer. But it turns out if you do have cancer, and we know what mutations you have, we can actually take that blood sample, look for those mutations, and it doesn't just tell us, yes, you've got some mutations in your blood that came from your cancer cells popping, like all cells do. But in fact, we can quantitate how many mutations we're seeing, and we know how much cancer you have and how much active cancer you have in your body. So as you may know, we do CAT scans and MRIs when we give somebody with cancer treatment, and we're looking for tumors shrinking, which means the volume of cancer is reducing, or we're looking for, unfortunately, cancer growing, which means our treatments aren't working. Well, all of that could be replaced by just doing a blood test every month or two and comparing the amount of tumor-based DNA you're seeing. And you could monitor the progress of your cancer therapy by the quantitative number of mutations you're detecting in your bloodstream. And this is what a liquid biopsy would be. And there's very promising evidence that now you can have a better sense of how a person's cancer is behaving with these liquid biopsies. In other words, they can give you a signal that the treatment's not working any longer, weeks or even months ahead of your standard CAT scans and MRIs. So in the near future, I would like to think that all our radiologists could retire and we could shut down our radiology departments and we'd just be doing blood tests and telling you, how the cancer that we know you have is behaving. Okay, so that's going to happen, I think, in the next handful of years. And I'm being facetious. Of course, we're still going to have X-ray machines and CT scans and MRs, but it may be just easier and actually cheaper to monitor the progress of your cancer with a blood test. So that's one kind of liquid biopsy. And I'm kind of excited about that because it would be simple and quantitative and pretty straightforward. The second part of the liquid biopsy that I think is even more exciting is that right now we have what we call screening tests for cancer to catch cancer early. So those screening tests are things like a mammogram for women and their breast cancers, for people who might develop colon cancer, which means basically everybody my age and older, you need to have a fecal occult blood test, a FIT test. And this means we hope to find an early cancer by detecting blood in your stool. Or you can have a colonoscopy every few years as well. So these are the kind of screening tests we do to pick up early cancers. Well, it turns out there's emerging evidence that you can actually do this liquid biopsy idea even before a person has a known cancer. And it's possible to identify that in your blood, there's an unusual number of, let's say, p53 mutations. And p53 is a mutant gene in about half of cancers. So many cancers, even before they're, obviously diagnosed, will be shedding little bits of this DNA that you can pick up because the DNA is not normal. It's floating around in your blood. It's left the tumor, and the tumor could still be small, but we can detect the presence of these unusual mutations in the blood. And this kind of liquid biopsy may actually become available as a screening test where you can detect early breast cancers, early colon cancers, and actually ovarian cancer. Looks like maybe one of the things we'll be able to pick up in your bloodstream before we would know it's there. Because currently in 2021, there's no good screening test for ovarian cancer. Most women, when they develop ovarian cancer, it's stage 3 or stage 4, and there's no way to find it ahead of time that's ever been shown to be helpful. So if we imagine 10 years, so 2031, I expect much of our cancer screening is actually going to be maybe it's an annual blood test that would cost maybe $400 or $500. And, if it's abnormal, you get a call from your family doctor saying, Roy, we're looking at your screening blood this year. And, there's some mutations there that make us think that you might have a cancer, an early cancer. So we want to do some imaging tests and we want to order a CAT scan. And that we might be picking these things up earlier before they're stage 4, before they're symptomatic, before they're incurable. So this is, I think, a tremendously promising thing. I'm going to talk about 2 more promising things and then open it up to questions, if that's okay with you. I think you've probably had people talking about immune modulation. And this means immuno-oncology drugs. And it's based on the observation that the cancers that become clinically evident, the common cancers that become clinically evident to patients, cause symptoms, cause lumps and bumps and cause trouble, have figured out how to escape the immune system. What that means is for them to actually have grown, that means they've got a bit of an invisibility cloak, the Harry Potter cloak of invisibility, so that the immune system doesn't know they're there. And they're able to grow and divide and spread, and they're unchecked by our normal way of fighting off infections and tumors. So by the fact that you have a cancer, by definition, it's essentially slipped through your immune system. Now, there are drugs that rev up your immune system. And these are the new immuno-oncology drugs. They've, been marketed in Canada now for about 8 years. And they're making a big difference in some common cancers like melanoma and, the occasional patient with pancreas cancer and other settings, like Roy had mentioned. We're seeing some benefits in people with lung cancer, some evidence in bladder, and maybe one subset of breast cancer as well might be sensitive to these. So these are relatively non-specific kind of immune modulations. So imagine you're in a car, and essentially what you're doing is, taking the brake off the immune system. And what happens is the immune system revs up, and sure, it may kill the cancer cells, but the immune system may do things that immune systems can do to otherwise healthy people. They can attack normal tissues and cause autoimmune disease. So things like, thyroid damage from the immune system. The immune system can rev up and damage the lining of the gut. So it's a bit like Crohn's disease or colitis. You can have immune attacks on the lungs, for example, and cause an autoimmune pneumonitis. So people can even develop, rheumatoid arthritis-like problems from these drugs. So it's very helpful. These drugs, improve survival. They control many cancers, and they're wonderful additions. Please don't get me wrong. I'm not complaining, but they're not perfect. Okay. And the reason they're not perfect is because when you take the brake off your car, you're going to rev, you're going to keep going. And, it's going to be unpredictable exactly where you're going to end up. Now, what I think is going to happen, and there's a lot of evidence coming this way, that the really most exciting, most specific ways that we're going to tweak the immune system is using a kind of therapy that is rapidly evolving, and it's getting some astounding results in some relatively rare cancers. But the potential is there for, in principle, any kind of cancer. And I've if any of you read the thing I wrote, I call them bespoke immunotherapies. So these are tailor-made ways of getting your immune system to attack your cancer. It's not your immune system to rev up and attack stuff. It's your immune system specifically tweaked to attack specifically your cancer, which is another huge leap above what we generally have today with immunotherapies. And the way this is done is you donate some blood, and your blood cells are taken out. And there's a kind of immune cell called your T cell. There's B cells and T cells that are the lymphocytes, or the white blood cells, one subtype of white blood cell in your blood. Now, these T cells have a job of going around and fighting infections, but you can reprogram them, and you can actually reprogram them to specifically attack the problem that your cancer has. So there are some really, really interesting early results coming from studies where you take a person's tumor, you study it molecularly, you find out what mutations it has, and you figure out what proteins it has that are abnormal. So the abnormal DNA leads to abnormal proteins, and those proteins are, in the wrong places. They're shaped incorrectly. They're the kind of thing that an immune system could attack if it were trained to attack it. And then, so you know that there's a P53 mutation. So the P53 protein is shaped incorrectly and malfunctioning in cancer cells, but not in normal cells. Then you take the T cell and you specifically train it to attack that abnormal P53 protein. And then you take that T cell that you've trained, and then you give it growth factors, and it divides into 2 and into 4, into 8 and 16. And you make millions of these specific immune cells. And then you re-inject your own immune system that's been retrained back into your blood. And then it goes into your body and it attacks the cancer cells. And in my mind, of all of the cancer therapies that one could imagine, this is the most promising. This is the holy grail. This is where we could be potentially curing people of advanced cancers by understanding their biology and taking one's own immune system and specifically training it to kill only those cancer cells that have that abnormal biology. And this is expensive technology. In my area, which is treating breast cancer, the first case report of this came out in 2018, and it came from MIT and Harvard. And that one patient, her treatment probably cost on the order of $2 million, perhaps a bit more than that, with all the molecular biology that was done and the reprogramming and all of the activities that went around that one patient. But to date, she had incurable advanced cancer, and she's alive and well as we speak today. Now, this is, I think, where the field is eventually going to head. And in my mind, this is incredibly difficult. The expertise is not everywhere. The experts are swamped trying to figure out how to do this, even for one or two or a handful of patients. But in principle, this is a technology that could be learned, that could be scaled, and potentially could be, the real optimal anti-cancer therapy for patients with advanced disease. And I think we're going to be there in a much bigger way than we are today. In 10 years from now, I think this is going to be available even in Canada, even with our limited resources, and even with our philosophy that we worry more about the group than one individual. So I think there's hope here as well. So I'd just like to wrap up and say that cancer clinical trials are a journey that the physician, with humility, takes with a volunteer patient who knows that they're treading into the unknown, that it's the only way that we are ever going to make progress against this horrible disease. That what we've done so far is great in that we've had some progress, but where we're at is by no means good enough. But there's still many reasons to be hopeful, because we're understanding things far better than we did in the past. And we're now coming up with the kind of tools that would give us a real chance to cure patients with advanced cancers.